Quite a bit of expectation, I guess. But anyways, thank you, Jerry, and thanks, Carol. And what I'm going to do now is I'm going to go into more basic mechanisms of inflammation associated with smoking and also inflammation associated with exacerbation of COPD. But before I start, just the conflict of interest. I do have research support from AstraZeneca and MedImmune, and I do have research support from Roche. I don't think it has any bearings on the presentation here, but just for completeness, I declare that as a conflict of interest. Now, before I go into the smoking and how smoke elicits inflammatory processes, I just want to step back for a moment and come to this statement here by the WHO, by the World Health Organization, which basically is that half the people that smoke today will die, will eventually be killed by tobacco. So if you are a smoker, you have a fairly high risk that tobacco smoking will eventually be the cause of your death. So if you're still smoking now, probably a good idea to step out of this lecture hall and quit smoking after, or seek help in quitting smoking. Smoking as such is responsible for about 90% of all causes of lung cancer, 75% of chronic obstructive pulmonary disease, chronic bronchitis, and emphysema, and it also contributes to, and that would be at the bottom here, to ischemic heart disease. About 25% of all ischemic heart diseases are related to smoking. But what I'm going to do now is I'm going to go specifically into COPD. And the objectives that we have for today, they're two very simple objectives: discuss mechanisms of cigarette smoke-induced inflammation. As we go through it, you're going to realize that it's actually not as simple, unfortunately. And then discuss the impact of cigarette smoking on microbial infections. And both of these studies are pursued in mice. And the reason why we're going to focus on inflammation is, by and large, and you recognize that slide down here, what it basically shows is that in smokers, you have an accelerated decline in lung function. And it's generally believed that inflammation contributes to this accelerated decline in lung function. So this, I think, justifies to focus the presentation today on inflammation. Now, what does inflammation do? If you look at a normal bronchi over here, it's nice and open, plenty of space to pass your air. In the context or in response to smoking, your airway is going to change in that you actually have a thickening, and you also have the accumulation of mucus in your airways. And it's fairly easy to understand that this will compromise the way you can breathe. And in here, the pathways, the inflammatory cells that I will focus on is on macrophages and neutrophils. Both macrophages and neutrophils are believed to contribute to this inflammatory process. Macrophages and neutrophils also contribute likely to the airspace enlargement. And if you look at a healthy human lung and you compare that to a smoker's lung, it's fairly easy to appreciate that something went wrong here. And what happens here, and there's good evidence for that, that proteolytic enzymes produced by macrophages such as MMP12 or proteolytic enzymes such as neutrophil elastase produced by neutrophils, they contribute to this airspace enlargement, where in a late-stage disease, by and large, the lung is no longer intact. It is largely compromised, and you can actually see through it. That's how far the lung is damaged. Now, the good news is we made tremendous progress on processes of cigarette smoke-induced inflammation, on inflammatory processes associated with chronic obstructive pulmonary disease. And here you see a review from Peter Barnes, where again, the macrophage is listed, you have the neutrophil involved in it, you have cigarette smoke that activates macrophages and the epithelium, which will now initiate an inflammatory response that eventually will lead to mucus hypersecretion, airspace enlargement, and something that Dr. Cox alluded to, also potentially the stiffening, the fibrotic changes that can be associated with COPD. So that was in 2008. 2009, the picture starts looking far more complex. Again, the good news is we're starting to understand the disease better. More and more mediators are being listed. There's also new cell types that start showing up, Th17 cells. There's even a mentioning of the eosinophil. So the picture gets more complicated, and the projection is by 2015, that will be the picture back then, or in the future. A rather complicated picture, and everybody that has traveled to London is recognizing it as the subway map of London, right? So the process, the mechanisms that anti-inflammatory mediators are being characterized, they start depicting a far more complex picture. Now, where do mouse models become useful? How can we now use an experimental system to study the relevance of given inflammatory mediators? And what we've done here is that, for example, we focused on interleukin-1. Interleukin-1, the first cytokine to actually be mentioned as interleukin-1. By now, we're up to interleukin-37, unless somebody in the audience is better informed than me. Sorry? 38 or 39, so we have discovered even more. Interleukin-37 belonged to the interleukin-1 family, so I was quite excited about that, but apparently my knowledge is outdated. So, interleukin-1. What is the importance of interleukin-1 to smoke-induced inflammation? The way we tested that, we utilized the Marlboro mouse, or a mouse exposed to cigarette smoke. For the younger ones in the audience, you've probably never seen the Marlboro Man. It used to be an icon of smoking, the advertisement, the cool Marlboro Man with his cigarette in his mouth. Our mice don't look as cool. They're exposed to cigarette smoke in a whole body exposure chamber. For quality control, to make sure that they reflect what is seen clinically, we looked at carboxyhemoglobin and cotinine level. Cotinine, the stable metabolite or breakdown product of nicotine. And if you compare what we see in animals to what we see clinically, the numbers match up quite nicely. So we seem to expose them to a level of cigarette smoke that appears to be, I would say, relevant. In animals now, we have the advantage that we can knock out specific genes. And what we used here was a mouse that was deficient in the IL-1 receptor. So it can no longer, IL-1 can no longer signal in that mouse strain. And what you observe in here now, if you expose an animal to cigarette smoke, you have a robust neutrophilic inflammation. If you do that over the background of IL-1 receptor 1 deficiency, you have an attenuation of the neutrophilia. So, smoke-induced neutrophilia is IL-1 receptor 1 dependent. By extension, yes, IL-1 is important in this process. And we observed that in the bronchoalveolar lavage, and we also observed that in the lung tissue. This is something that we're not the first ones to show that. It has been reported before, so the observation is reproducible. A group out of France reported that smoke-induced inflammation is IL-1 receptor 1 dependent. What is interesting is that the IL-1 receptor 1 dependency is specific to neutrophils. We still have an increase in mononuclear cells, that being macrophages and likely monocytes that infiltrate into the lung. The relevance now to airspace enlargement, IL-1 receptor 1 is involved in airspace enlargement. It's a study out of Vancouver by Andrew Jerk, where he showed that IL-1 receptor 1 knockout mice are protected by approximately 65% as it pertains to emphysema formation. So IL-1 receptor 1 appears to play an important role in smoke-induced inflammation, but also in the formation of emphysema. If we now look mechanistically at the rather complex graph here, where we have a number of different mediators, and we focus specifically, for example, on TNF-alpha and other inflammatory mediators, a number of chemokines, and matrix metalloproteinase 12. The matrix elastase produced by macrophages that contributes to airspace enlargement. Is the induction of these mediators IL-1 receptor 1 dependent? And the answer here is yes, they are all downstream of IL-1. So when we went further on, and I'm just gonna go to the graph, the bottom line of, we did experiments with chimeric mice, with interventions using antibodies that blocked IL-1 alpha and IL-1 beta. The bottom line is that macrophages elaborate IL-1 alpha that now signals on epithelial cells. Epithelial cells then elaborate inflammatory chemokines such as CXCL1, 2, and 5, leading to the recruitment of neutrophils. Neutrophils do carry IL-1 alpha on the cell surface. They can signal back as a feedback loop, and as inflammation progresses, there will be then tissue destruction. So that was quite simple. Smoke-induced inflammation is IL-1 dependent. How come clinically we have a very limited clinical armamentarium? And the answer now here, in animal models, we take a reductionist approach. These are inbred mice. The smoke exposure is standardized. The reality is very, very different. Cigarette smoke, every individual is different. That's what Dr. Cox referred to as the audience. They're all different genetically, and they all smoke different. So what we have to expect is a certain heterogeneity. And these pictures, I just random downloaded them from the internet. If you see yourself on here, then I profoundly apologize. Anyways, all smokers are different. The genetic background is very different, meaning that there is cigarette smoke and there is differences in genes and environment that will predispose certain individuals to the development of COPD. On top of that, the combination of cigarette smoke, genes, and environment may predispose certain individuals to microbial colonization. And at this point in time, we actually do not understand how that contributes to inflammatory processes. So the picture that I was drawing with smoke and IL-1 was probably a bit oversimplified. And on top of it, there's a big aspect with the aging. With aging, there's also inflammatory processes associated with that may contribute to disease progression. By and large, the reality has it, when we look at inflammation associated with COPD, it's not as simple as cigarette smoke, activating macrophages, and then signaling to the epithelium to actually elaborate inflammatory mediators that will now lead to the recruitment of neutrophils. As a matter of fact, the processes are far more complex. There's also chronic innate immune activation that may lead to a breakdown of self-tolerance. There is an emerging literature that smoking may actually propagate or is associated with autoimmune processes. At this point in time, an interesting concept that still needs to be tested and proven. There's also issues associated with the colonization that I mentioned, but what I really want to focus on for the rest of this presentation is on how cigarette smoke affects viral and bacterial infection. So again, I'm going to take a reductionist approach, where rather than mirroring the whole complexity of COPD, we're just going to investigate how does cigarette smoke now affect viral and bacterial infection. Now, why is that of relevance? It is of relevance because, as it was mentioned before, the etiology of COPD exacerbation, 75% of all exacerbations are related to either viral, bacterial, or viral and bacterial co-infection. So 75% of all exacerbations are associated with microbial exacerbation. So, what is now the issue in COPD? Why do they have those exacerbations? And in here, I'm going to come back to the little puzzle scenario where disease severity. The more severe the disease, the more frequent the exacerbation. Now, why is that? And there may be a very simple explanation for it. It's likely oversimplified, but what we have in our daily life is there's normal fluctuation in lung function. What you appreciate that if you're young and healthy and you still have plenty of lung function left, that daily variation may actually not bother you at all. But as you decline, and if you're a smoker that is susceptible to more rapid decline in lung function, then you appreciate that even daily variations may actually lead you at the brink of disability. And as lung function further depreciates, you end up in a situation where even something such as a daily variation in your lung function may bring you at the brink of death. So the decline in lung function, the disease severity, may actually be a very important contributor to the frequency of the exacerbation. At the end of the day, it may actually be a numbers game. How much lung function do you have left? And how much of a decline in lung function can you afford with a viral or a bacterial infection? Now, of course, if you continue to smoke, you have more frequent and you also have more severe exacerbations. Another factor playing in, again, are the microbial colonization. Here I would like to tell you what the relationship is. At this point in time, there is an association between microbial colonization and frequency of exacerbation. On a molecular level, we actually have very little understanding how the two relate to each other. And last but not least, just to keep everybody happy, there are some individuals that probably have a genetic predisposition to have more frequent exacerbation. This is an area that we still know far too little about, and that I hope that the future will hold some answers. But what I'm going to focus now is specifically on how does smoking impact a viral or a bacterial infection. I'm actually going to specifically focus on how smoking affects a viral infection. What is important here is that there is good epidemiological data to suggest that both upper and lower respiratory tract infections are more frequent even in young, otherwise healthy smokers. So smokers have more frequent upper and lower respiratory tract infection. So the problem that they face dealing with microbial infections may not just be when they actually develop COPD, it may be something that they have inherently, even at a younger age. So, what are now the issue? Is it that they're more susceptible? Is it that they can't clear the pathogen? Or is it that they just respond more robustly? Or that they respond with a greater inflammatory response to those agents? And again, as I said, I will focus mainly on viral and bacterial and viral infections. I'm more than happy to discuss in the question and answer period the consequences to bacterial infection. But with a viral infection, the way we modeled it here, we exposed mice to cigarette smoke, here only for 4 days. We did the same experiment with 8 weeks. We've done similar experiments with 24 weeks. By and large, the picture that emerges is very, very similar. By and large, with cigarette smoke, an influenza infection, and here we utilized a mouse-adapted virus. What you observe and what you appreciate is a smoke-exposed animal mounts a far more robust inflammatory response. And this is not something that is unique to our own lab. We published it originally in 2006. It was subsequently confirmed by a group out of Australia and a group out of Yale to basically reporting that cigarette smoke exaggerates, or smoke-exposed animals have an exaggerated response following infection with influenza. So it is highly reproducible. Several different labs across different continents have had the same observation. What was interesting, and that's a study out of Yale, what's interesting here is that they showed, in addition to the exacerbated inflammatory response, they also looked at markers of emphysema formation. And what makes that study particularly compelling and interesting is they actually provided clear experimental evidence that viral infection can contribute to the airspace enlargement. What is interesting is that we looked long and hard whether cigarette smoke compromises the ability to fight off the virus. Is there an increased viral burden? Is there a decreased or a delayed viral clearance? And as a matter of fact, under our experimental conditions, the mouse is able to handle the virus quite normally. What's different is they just mount a far more exaggerated inflammatory response. And when we went after it in terms of molecular mechanism, then the picture that came out is the exacerbated inflammatory response was IL-1 receptor 1 dependent, where linking back to what I said before, cigarette smoke through an IL-1 alpha dependent pathway elicits a neutrophilic inflammation. If you now superimpose a viral infection, then the viral infection, where the epithelium is already activated, leads to a greater release of inflammatory mediator and consequently a greater accumulation of neutrophils. And if you now take that into the context of the study out of Yale, then this has the ability to cause more airspace enlargement. We don't know yet about the role of IL-1 beta. That's just to be complete. What emerges as a picture here is that if you have a non-smoker and a smoker, as they go through their daily lives, as they go through life, the smoker will have repeated bouts of more inflammation in response to viral and bacterial agents, which will induce more damage. The good news is we have some capability to repair damage, but not every repair is perfect. And eventually, what will happen over time is that a smoker will cross an imaginary threshold for disease expression far earlier than a non-smoking individual. So what we're basically postulating is that the altered response to viral and bacterial agents is contributing to the accelerated decline in susceptible individuals. With that, there's one last slide, likely the most important slide. Experimental research in the lab is a cumbersome effort. There's a lot of people involved in here, and it wouldn't happen without a very talented group of postdocs, grad students, undergrad technicians, as well as a number of collaborators in Canada, in the US, in Japan, in industry, but also in academia. So none of that would happen without the team here that can work together. With that, I'm more than happy to answer any questions.